This is my ETS, electron transport system, oxidative phosphorylation, chemiosmotic model video tutorial. And at this point, we're going to pick it up, aerobic cellular respiration, following glycolysis, following matrix reactions, and going into the third stage of aerobic cellular respiration, and looking at the electron transport system. So what I've got here is a diagram of part of the mitochondrion, and I'll make this diagram available to you basically as you can see it, minus some of the navigation tools here and minus the clipboard. But all of this stuff will be available for you to download from my website. I'll put the link in the information box. So if you want to go ahead and get that and print it out, you can follow along pretty much exactly what I'm doing on this tutorial. All right, so what we have here really is the mitochondrion, and this area here is the intermembrane space, so that if we were looking at a mitochondrion right here, and you don't want to draw this on your worksheet, by the way, but if that was a mitochondrion, that would be the outer membrane, and I'm drawing the inner membrane with all of its folds like that. And so this intermembrane space is this area right in here. That's the intermembrane space. And then, of course, there's a phospholipid bilayer on the outside, and that's this part. We're only seeing a little bit of the phospholipid bilayer, or the membrane, the outer membrane of the mitochondrion. That would be right there. And then this wavy line here represents the inner membrane, but I didn't draw it like that. I just drew it straight. All right, so that's the phospholipid bilayer. That represents the inner membrane of the mitochondrion, and then into the matrix where Krebs cycle occurs and the pyruvate oxidation, the matrix reactions, and that would be down here. All right, so we've got the matrix region down here. We've got the inner membrane of the mitochondrion, the outer compartment or the intermembrane space, and then the outer membrane, just to sort of orient you. Now, embedded in the inner membrane of the mitochondrion are a bunch of molecules, complexes, proteins, that kind of thing, that make up the electron transport system. Now, there's also this thing over here, which is not really part of the ETS, but this stuff here is the electron transport system, or the electron transport chain. We've got a molecule here, and we've got one here, and here, and here, and here. And you don't want to be adding these numbers. We're just going to be looking at names of these structures and the path of electron flow. Now, this thing over here is associated with the electron transport system, and it's the part that makes the ATP. And of course, there would be many of these groupings throughout the inner membrane of the mitochondrion. It's not, you don't just see this stuff just one time. It's all over the cristae, the folds of the inner membrane. It's all throughout here. There'd be one there, and another grouping there, and another grouping there, and there. So it's not just this one thing. All right, so let's identify some of these components. Now I'm going to come down here, turn the names on. Now, if you do have this printout, and if you can find the room to write the names, or if you decide that you would rather do some form of numbering, you know, you could call this number 1, you could call this number 2, and number 3, number 4, number 5, number 6, and maybe number 7. Or you could start at 1 if you know, something like that. You could do that if you didn't want to write, you know, the names right beside them if there's not quite enough room, you know, or you could write them down here, something along that line. But what we have here is something, first of all, called the glycerol phosphate shuttle, okay, and that's this thing, and I'm going to talk about that in a moment. This shape right here is called NADH dehydrogenase. We'll be talking about that. Down here I've labeled Q. This is also sometimes called coenzyme Q or ubiquinone, but I've just put the short form Q. This is cytochrome BC1. This is a complex, so a lot of these things are made of different parts, you know, like a puzzle piece of components. It's a lot more complicated than as drawn, but that's cytochrome BC1. This is cytochrome C, and this is cytochrome oxidase. All right, so we're labeling these big blob-like structures, these things that are part of the ETS and this shuttle over here. And then we have a hydrogen ion pore. That's this thing, which is like a facilitated diffusion channel. And we've got an associated enzyme here called ATP synthase. So this blob would represent that. This is an enzyme that's going to create the ATP. In addition to that, we've got Krebs cycle down here. So you can see an arrow coming down here, churning off some kind of product as Krebs cycle occurs. All right. So we're going to write some names into these boxes here, and then I'm going to show you how this whole thing works. So we might as well start over here. Glycolysis, which breaks down glucose and produces 2 pyruvates, also produces ATP, 2 ATP per glucose, and 2 NADHs. NADH is an electron carrier. It started as NAD+, and it picked up 2 electrons and some hydrogen, and it was converted into NADH. So this molecule, an electron carrier, has high energy electrons that were taken from the glucose. These electrons on NADH cannot get through the inner membrane on their own in some tissues. And in those tissues, you need a shuttle or a carrier, and it's called the glycerol-phosphate shuttle. And the NADH is actually transferred to glycerol phosphate, and that then transfers those electrons through the membrane to a molecule of FAD. Now, FAD is an electron carrier as well, and when FAD picks up electrons and some hydrogen, it is converted to this, and I'm going to write this in the box. So the most important stuff is the stuff that I'm writing in the boxes, and all of this, some of this extra stuff, anyhow, you don't have to write. You're welcome to. There will be some stuff over here that's a good idea to do. So we get the FADH2 from all of this. All right, so that's what we really care about, FADH2. But you've got to remember that what we've seen is that NADH has been converted into FADH2 through this sort of handoff. Now, that will be important, and you'll understand why in a little bit. So we've got some FADH2. In addition, of course, the pyruvate from glycolysis goes through the pyruvate oxidation step of the matrix reactions, or the pyruvate oxidation or preparatory steps. And that pyruvate is converted to acetyl-CoA, which goes into the Krebs cycle right down here and goes around. But the preparatory step I was just talking about, the pyruvate oxidation, occurs right here. And that produces some NADH. And we're not looking at really how many NADHs or FADH2s are being produced yet. We're going to account for that in a bit. But if you know this step, you'll know that some NADH is made here. All right, 1 NADH per pyruvate, 2 pyruvates per glucose, so there's really 2. But we're not going to worry about that right now. In Krebs cycle, at various points, there is more NADH made. There's some made here, and there's some down here, and there's some up here. And so we're going to write NADH also in this box. Okay, just clean up a little bit of that stuff. All right. In addition, Krebs cycle itself produces some FADH2. Of course, it's not here in the cycle. It's produced right around there, around fumarate, but we're just writing them off to the side. And by the way, this diagram is following roughly the diagram from the Nelson Biology 12 textbook, although I've created some new graphics here. But this is the way that we're going to represent them, with them off to the side, not coming off Krebs cycle at the exact position. Okay, we've got these electron carriers, very important. We also have some carbon dioxide coming off. This is going to be exhaled. This can leave the mitochondrion by simple diffusion. And down at the very bottom, Krebs cycle produces ATP. 1 ATP per pyruvate, 2 ATPs per glucose, because there are 2 pyruvates, but we'll account for that ATP a little bit later on. Okay, now we're basically ready to start, okay? The mitochondrion is the powerhouse of the cell. You've probably heard that before. And we can see a little bit of ATP made by Krebs cycle. All right? That certainly doesn't seem like a heck of a lot. The other ATP is going to be generated by these electron carriers as they drop off electrons and they travel through the ETS, the electron transport system. As those electrons move, a lot of ATP is going to be produced by the electron transport system, and that is why the mitochondrion is considered the powerhouse of the cell. Remember, glycolysis only gave us 2 ATPs, and we'll come back and talk about them in a bit. All right? And really, the matrix reactions only gave us 2 ATPs, 2 pyruvates coming through, 1 each, so 2 ATPs. We haven't made very much. We are about to make a whole lot. All right? So now, let's follow the path of these high-energy electrons by these electron carriers through the ETS. Let's start with NADH. All right? Now, NADH is going to float over and interact here with the first electron transport system carrier called NADH dehydrogenase. It's called this because it's going to dehydrogenate the NAD. It's going to remove the hydrogens or release the hydrogens and the electrons. And when NADH interacts with this complex here, it's going to release 2 electrons into the ETS, onto this first carrier. So imagine it bumps in, and here are the 2 electrons moving, along this carrier, and it's going to follow this dotted path. When those electrons are picked up by NADH dehydrogenase, some energy is released by those electrons. So I'm just going to draw a dotted arrow, and I'm going to say E for energy. Now, that energy released by the electrons is going to be used to pump hydrogen ions that are sitting here in the matrix of the mitochondrion. They are going to be moved through this complex, NADH dehydrogenase, and I'm going to write H plus right up here. They are going to be pumped or pushed into the intermembrane space of the mitochondrion. So from the matrix in the middle to the next chamber right here. Now, up there would be out of the mitochondrion. They're not going out. They're building up in this intermembrane space. That is going to increase the H plus concentration out here. All right? So we're pumping them, and we're going to pump more soon. So we're building up a concentration. So this is going against the concentration gradient, which takes energy. This is not facilitated diffusion. It's a pumping. The energy to do this does not come from ATP, as you may have thought. The moving of molecules, against the concentration gradient normally takes ATP. This doesn't. The energy comes from the movement of the electrons through the ETS. Now, we have pushed 1 hydrogen ion from the matrix to the intermembrane space due to the movement of electrons. Those electrons will continue to move down to the next carrier. This one is called Q. This one can float around in the phospholipid bilayer, pick up the electrons like a ferry, and carry them over to the next complex, like that. Now remember, every time a complex or molecule receives electrons, it's being reduced, and the molecule giving up electrons is being oxidized. So in this first step, NADH dehydrogenase is reduced by gaining the electrons. NADH is oxidized by losing those electrons. When Q picks up the electrons from NADH dehydrogenase, it is reduced. NADH dehydrogenase is therefore oxidized. So this is a series of redox or oxidation reduction reactions. All right. So the electrons have moved from Q to the next complex here, is called cytochrome BC1. When cytochrome gets those electrons, it's reduced. This one's oxidized. Energy is released again, and that energy is used to pump hydrogen ions from the matrix through this complex into the intermembrane space where the hydrogens are building up. So this is the second hydrogen ion pumped out due to the movement of electrons. The electrons then continue. They're losing some of their energy. They move up to this carrier. This one could actually float around as well. It'll come down here, pick up the electrons, ferry them over to this one. So now the electrons will move down to this last carrier or component of the ETS called cytochrome oxidase. This complex, when the electrons get there, releases energy. Let's do it up here this time. Energy is released, And that's enough energy for hydrogen ions that are floating around here in the matrix, and we could talk about where some of those come from in a bit. Hydrogen ions are pumped into the intermembrane compartments. So don't say out of the mitochondria. The intermembrane compartment, the intermembrane space, it's also sometimes called the outer compartment. That's our third hydrogen, all right? By that time, the electrons that originally came from glucose, all right, and were dropped off here and traveled through releasing their energy. By the time they get here, they have given up their energy, and now they are going to come off of cytochrome oxidase, and look at the name, oxidase. It has to do with oxygen. Down here, there is 1/2 O2. Now, that's the way we tend to write it. It really means it's an oxygen atom on its own, and a couple of hydrogen ions that are floating around in the matrix. Remember, there was a few to be pumped out, and there's a few here. And those electrons will then go onto the oxygen. 2 electrons will go onto the oxygen. All right. Now, if you can imagine, if oxygen, just one oxygen here, was to gain an electron and an electron, right, it's going to have a 2- charge. Well, take the 2 hydrogen ion charges, put them together, you'll get no net charge, and you'll get 1 O and 2 H's, and that makes water. So water is produced at the very end of the electron transport system because those electrons go on to oxygen. Oxygen is very important here. Oxygen is called the final electron acceptor in aerobic cellular respiration. And this is why you see the reaction C6H12O6 plus 6O2 producing water, etc., etc. Okay? The point I'm trying to make is there is the oxygen that you need for cellular respiration. It's right here. Oxygen is the final electron acceptor, and this is why you have to breathe. If you were to stop breathing or be in an anaerobic environment or situation, strenuous exercise, whatever it is, if oxygen is not available anymore, the electrons that come down the ETS get to this point and they have nowhere to go. Well, electrons are negative, and negatives repel negatives. So as more electrons start to come down here and have nowhere to go, they start to repel each other back, and then they can't come down anymore. Well, if they can't travel all the way through the ETS, and this will just back up right to here, all right? If it stops right here, you can't pump hydrogen out. Now, why do we care about that? Well, because these hydrogen ions being charged, they cannot simply diffuse through the phospholipid bilayer, right? They are charged, they are polar. The phospholipid bilayer won't let them through, and there's no way for them to get through except this. So at the right time, as the hydrogens build up, the hydrogen ions come over here, and they go one at a time through this hydrogen ion pore. This is facilitated diffusion. They're not being pumped. They're using the energy from the concentration gradient that is built up. They then go streaming through this tunnel-like pore, interacting with ATP synthase. Let's draw a big blob there. So imagine an enzyme there. As they come through and hit that enzyme, that gives a bunch of energy to that enzyme, all right? It's spinning around now. It's activated. And the movement of hydrogen through the hydrogen ion pore, interacting with ATP synthase, activating it, there's the hydrogen coming through. The energy of that is enough to take inorganic phosphate that's floating in the matrix and ADP that might collide normally but not have enough energy to stick together. But with the electron coming through, activating this enzyme can then take these two and push them together with enough force to create ATP. So now we're creating ATP in the electron transport system, and it's all because oxygen is available at the end to pull the electrons out, to give them a place to go. Think of it like a magnet, pulling them out, allowing new electrons to fly through this thing really quickly, pumping out hydrogen, which then comes through the channel. Now, there are 3 hydrogens that were pumped out. So when this one goes through, it creates 1 ATP. But then this one goes through, and we create another ATP. And then this one goes through. I know that's getting messy. And we create another ATP. So every time one hydrogen ion travels through the ATP synthase, hydrogen pore, and by the way, this thing collectively is sometimes called the respiratory assembly, the respiratory or respiratory assembly, because it deals with cellular respiration. So every time a hydrogen ion goes through, 1 ATP is made. So what this means is that every time NADH over here drops off its electrons and they go through here, 3 hydrogen ions are pumped into the intermembrane space. They go through this hydrogen pore, stimulate ATP synthase, 3 hydrogens, 3 ATPs. So every NADH created from the preparatory step we saw here, from Krebs cycle here, same thing, electrons going through. Every single NADH leads to the production of 3 ATPs. So we're going to have to count for all this ATP. That right there, that 1 ADH making 3 ATPs, that's almost as much as Krebs cycles 2 and glycolysis is 2. So you can see we're going to create a lot of ATP here. Now, I'm going to erase that for the moment. So hopefully you've got the information in the boxes and maybe a couple of other arrows and the hydrogens in there and the concentration. Okay. Now I'm going to erase that. And I want to say something about this NADH here. So I'm going to come back. I'm just going to write NADH back in this box. It would be the same for this one, all right? When this NADH drops off its electrons right here, it also releases the hydrogens, all right? So it releases its H+. Well, this could be one of the H pluses that gets pumped out, all right? This could be one of the H pluses that is involved with the formation of water. Okay? But the other thing is the NAD plus that this all started with is regenerated. NADH, without its electrons, without its H plus ion, is NAD plus. We have oxidized NADH back to its NAD plus form. This can now go back into either Krebs cycle or the preparatory step right here. So that is necessary to keep Krebs cycle going. And this is why Krebs cycle, or the matrix reactions, is considered to be aerobic, part of aerobic respiration, right? Matrix reactions, or Krebs cycle, let's say, and electron transport system, both part of aerobic cellular respiration. Krebs cycle, preparatory reactions, matrix and collectively, indirectly requires oxygen because it needs the oxidized electron carriers to continue to function. And if it continues to function, it makes more NADH. All right, so more NADH here. This one would do the same thing in terms of dropping off its electrons, making ATP, and being oxidized back to NAD plus and giving us some hydrogen ions. All right, so this is why oxygen is needed in the matrix reactions. Now, I also want to talk about FAD. So I'm going to switch to red. We have some FADH2 right here. Now, we also have some FADH2 over here. Okay, we'll talk about that one in a moment. Now, notice the dotted line. Look at where it leads. FADH2 is carrying electrons, but it doesn't go to the first electron transport system carrier, NADH dehydrogenase. And look at the name, NADH dehydrogenase, not FADH2 dehydrogenase. Instead, FADH2 comes up here, misses the first carrier, and drops its electrons off at Q. Because of that, this hydrogen ion is not pumped out when FADH2 drops off its electrons. Okay? They start at a later position. The electrons will now move along down to here, releasing energy. One hydrogen ion will be pumped out here due to the energy released by the electron or electrons. They'll move up here. They'll move down here. That's enough energy released to pump out another hydrogen ion, and those electrons will continue down and join on to the 1/2 O2 and the 2H pluses, and water is made. Diffuses out by osmosis, no problem. The net result, however, is that FADH2 leads to the production of 2 hydrogen ions. Now, I know you probably labeled the hydrogen ions and may have written the numbers, and so writing the numbers again would just be very confusing for you. But just don't worry about that. Just follow what I'm doing here. These 2 hydrogen ions, instead of 3, then will diffuse through the respiratory assembly, stimulate ATP, and ATP will be made. Again, it's 1 hydrogen for 1 ATP, but we've only got 2 of them now. So that means that we only make 2 ATP for every FADH2. And one way that you can remember that is FADH2 has a 2 there. All right, it's too bad NADH didn't have a 3 there. But okay, so NADH leads to the production of 3 ATP because 3 hydrogen ions are pumped into the intramembrane space and then re-enter the matrix this way. Each FADH2 that is produced leads to the production of only 2 ATPs. The same thing goes with this FADH2 over here. Now remember, it started as NADH from glycolysis, and if you need to, you go back and you see the pathway, glucose all the way down to pyruvate. Somewhere along there, you make an NADH on that side. You make another NADH. You've got 2 NADHs. You would think they would make 3 ATP. But in some tissues, and in particular in the one I've drawn, the one that has the glycerol phosphate shuttle, those NADHs are passed to FAD in order to get in. Think of it like there's a fence here, and NADH is on one side and FAD is on the other. There's no NAD plus around, and it's like I'm going to pass my electrons through the fence, and it's this guy that picks them up, not NAD plus. And so because FAD's there, it makes FADH2. This stuff does the same thing. It goes in just like this FADH2. So the glycolysis NADH can produce FADH2, which leads to the production of 2 ATPs. All right. Now, by the way, let's not forget that when FADH2 drops off its electrons right here, it will then release its hydrogens. They're now available to be pumped or to join with water. And FAD will be created. This is the oxidized form back into the matrix reactions, into Krebs cycle. All right. So the oxidized carrier, again, requiring oxygen indirectly to pull the electrons out of the ETS in order to regenerate this so that Krebs cycle can continue. Without this, Krebs cycle will stop, right? It'll stop there. It'll all back up. It'll all back up all the way here. It's done. Without these, Krebs cycle will stop. Without the NADH here, this will stop. All you'll have left is glycolysis, and it won't be able to pass its NADH into here. And by the way, when it passes its NADH into here, guess what happens with that? Back to NAD plus, back into glycolysis to keep glycolysis going. All right? If you can't drop your electrons off here, you can't reoxidize the NADH. We need another pathway, and that's called fermentation. All right? That's a whole other ballgame. So let me see. What have I left off? I should have labeled pretty much everything. We need to tally up all of the ATP being produced. This is a pretty complex situation here, right? So let's do that. All right, I'm going to come down here and I get my little clipboard going, open that up, and let's have a look at the reaction versus the electron carriers and how much ATP is made in the end. Now, if you like to, you could always write this on a note, a separate note, or on the back of your diagram or something like that. So we're going to look at glycolysis, and we are going to look at Krebs. Probably should have called that matrix reactions. Let's do that. Okay, let's call this matrix reactions. Okay, so that we include the pyruvate oxidation and that kind of stuff. Okay, so now glycolysis produces 2 NADHs. All right, but in this example, the NADH passed its electrons over and produced FADH2, which led to the production of 2 ATPs. So I'm going to put in brackets times 2, meaning each NADH produces 2 ATPs because they're converted to FADH2s. So that gives us 4 ATP from the NADH from glycolysis made by the ETS. Now, glycolysis also makes ATP on its own. It makes 2 ATP per glucose, and we're done. The matrix reactions, if you include the preparatory steps and everything going on here, there's an NADH produced down here, there's an NADH here, and there's another one over there. 4 NADHs per pyruvate, therefore 8 NADHs per glucose. So we're doing this per glucose. So there are 8 NADHs carrying those energetic electrons, and NADH produces 3 ATP per NADH. So I'm going to go times 3 here. That gives us 24 ATP. Now you're starting to see why the ETS is the real powerhouse. All right, but that's not it. There's an FADH2 produced down here. But because we're dealing with 2 pyruvates, there are 2 FADH2s. Although they only produce 2 ATP each, that's another 4 ATP. And let's not forget that down here we had some ATP, 1 per pyruvate, so a total of 2 ATP without any electron carriers. Just like that. Okay? So what does that give us as a grand total? We have 4, 6, 30, 34, 36. 36 ATP molecules produced, and this is per glucose. All right? So, recall when we went back to glycolysis and we had just looked at that reaction on its own, it was making just 2 ATP net. All right? And that was it. And in anaerobic conditions, you're not going to be able to use the NADH as I showed you up here. So you're going to get 2 ATP. So obviously something else has to exist to make more energy in anaerobic conditions. But if oxygen is available, that stuff that you need to breathe, this is what you can get. 36 ATPs per glucose. Now that's just one glucose. Imagine that you are metabolizing a starch molecule or a glycogen here with, hundreds of glucose, each split by, the enzyme to release the glucose. Think of all the ATP that you're making every second of the day. All right? Well, that is how the ETS works with all of its components. Keep in mind, there is more to this. This is my little quick summary, and I hope that helps you understand it. Thank you very much for watching. See you later.